Hello and welcome. In this third video on genetics, we are going to look into the topics and the potential question of biotechnology or applied genetics. We are calling it applied genetics because essentially they are application of molecular biology that we have studied until now. There are a few topics which have oft been repeated in the examination, but each one of these topics are equally important because if you see the last 11 years question paper, you will realize that each one of these topics have been asked in the examination either as a short note or a comparison or in some other form in the last 11 years paper. The topics are pretty scattered in the genetics syllabus. They are intermingled with transmission and traditional molecular genetics. Therefore, I have identified and I have isolated all these topics at one place, and then I have reorganized them according to a logical sequence of study. In this video, we are going to see all of this. Apart from that, in this video, I'm also going to address an important issue, and that is a number of good genetics textbooks do not do a good justice to biotechnology topics. It will be best to rely on certain specialized books on biotechnology to do a good coverage of these topics at hand. If you are not having access to these books, I will tell you certain shortcuts also, but first let's take a look on the topics themselves. Once we have seen the topics and scope of the topics, in the end I'm going to discuss about what are the books that you should be studying for these topics. So first and foremost, take a look on the topics which are given, and let's go full screen mode so that I can explain it a little better. So this is how the topics are arranged. As I told you that the topics are haphazardly scattered among the topics of molecular genetics and also certain topics of transmission genetics. But if you isolate the topics, there are in total 11 topics which you can see on your board. But in my opinion, the correct order of the topic should be slightly modified. So here I have identified the same 11 topics, but I have reorganized in a little logical sequence of coverage. When you cover these topics in this sequence, you will be having a conceptual flow and you will be building a good understanding of all these topics. Needless to mention, a good understanding goes a long way, giving you the ability to write balanced answers with certain good facts example. But everything starts with the soundness of your understanding. Therefore, please try to follow this sequence of coverage of biotechnology or applied genetics topics. It starts with DNA cloning, then it comes to recombinant DNA technology. Once we have done these two topics, you will be really ready to understand a little in detail about the different types of vectors, that is plasmids, cosmids, and artificial chromosomes as vectors. In this category, you will be studying a little more closely about plasmids and then about cosmids because they are two of the most widely used categories of vectors. But artificial chromosomes are fast emerging as an important class of vectors, especially when you have to send a large amount of DNA to certain target cells. The artificial chromosomes can carry such humongous amount of DNA which are almost unimaginable to be sent either using the plasmid or cosmid vector. Therefore, we have to study artificial chromosome. They are emerging, and apart from that, they are pretty much a part of the syllabus, so we have to study them. Later, we study transgenics. Transgenics is that discipline of applied genetics which also includes parts of developmental biology, and in this we are going to study how do we raise transgenic organism. In the context of zoology syllabus, we are actually going to study how do we raise transgenic animals, what are the methods, what are the steps in individual methods, and eventually what are the motives for developing transgenic animals, what are the limitations, and what are the benefits that we expect to get from animal transgenics. Interestingly, this is also a topic in economic zoology. So when you do transgenics here, you do not need to do transgenic there. So once you have covered the scientific basics, in this part only you are going to study about transgenic animals, their examples, their applications, and that will be sufficing for a question in economic zoology in paper 1 also. In zoology syllabus, there are a few topics which find their mention at more than one places. Transgenics is one of them. After that, you will be doing whole animal cloning, and the syllabus says you have to study its principles and methods. We are also going to dig deeper into what are the types of cloned animals and how do we go about producing a specific type of cloned animal. After that, we will be studying RFLP, RAPD, and AFLP. The next topic is application of RFLP in DNA fingerprinting or DNA profiling. RFLP is one of the most commonly used marker when we are going about DNA profiling, and the topic specifically asks you to study the application of RFLP into DNA profiling process. After that, you'll be studying genomics, and after genomics, you will be studying human genome project. First, genomics, its methods, its objectives, and how do we go about it. After that, you will be knowing about the project which aimed at mapping the entire human genome and then identifying all the genes, the nucleotide sequence of every single gene. Then the project aimed at determining the function of every identified gene, and to a great extent, the project met all its stated objectives. We are going to study that as a later part of this section. After that, you will be studying Ribozyme Technology. Ribozymes were discovered in middle of 1980s by two different scientists, and since then, first the basic notion that all the enzymes are polypeptides or protein, that basic notion got challenged. Later, we started discovering a number of applications of ribozymes, and today we have Ribozyme Technology as a part of our zoology syllabus, and we are going to see what are the questions which can be asked to you on Ribozyme Technology and what are the dimensions of Ribozyme Technology that you ought to be studying for your examination. Last topic is Proteomics, which is essentially the study of the proteins in molecular details, and it also includes in its later parts comparison and studying the protein families and their evolution. The very first topic here is DNA cloning. When you study DNA cloning, first and foremost, try to understand the meaning of DNA cloning. DNA cloning is a process, whether it is natural or it can take place in vitro, by which we produce copies of a DNA segment which has a well-defined nucleotide sequence. That's what DNA cloning is all about. In nature, DNA cloning takes place when genome replicates. So every cell clones its DNA before the cell divides into two daughter cells. This is actually a necessary prerequisite of cell division. You have to study what is the proper meaning of DNA cloning. Definitions are very important because once you have a compact but covering everything kind of definition, it becomes an important asset to be used in most of the answer types. Most of the answers in a subject like zoology, or for that matter even in other biological science optional like botany, most of the answers have to start with a good definition. A definition is something that builds your early impression in the eyes of the examiner, and therefore your definition stock should actually be of very high quality. For this, you need to study from good books and not from any and every book, and by the same logic you should not be relying too much of just any internet source. Authentic book has never any substitute. Having said that, once you have studied about DNA cloning, the next topic is that you should be knowing a little about what is a gene library and what is a genome library. Interestingly, when I teach in the class and when I ask my students about the definition of genome library and gene library, most of the students presume that it is basically a physical collection of specific genes in many copies, or it is a physical collection, that is in vitro collection of the entire genomic material in some kind of preserved setup. Interestingly, neither of these is correct definition or correct explanation of what's a gene library or what's a genome library. Find what is a gene and genome library from a good book. Some of you who come from biotechnology background or who have studied biotechnology carefully during graduation might already be knowing what I'm hinting at. But for the rest of you, it is a challenge to explore it in good book and find what is the correct meaning of gene library and genome library. If you are not already knowing it, trust me, you are in for a surprise when you discover what really is a gene library and what really is a genome library. So go ahead and find it out. But you need to build this concept, and these are the foundational areas to help you write a good answer on gene cloning. After that, you will be coming to cloning proper. There are two types of cloning, as you can see. Cell-free cloning, essentially how you create different copies, so many copies of the same DNA, that is cell-free cloning. One of the fastest way to obtain cell-free cloning is nothing but polymerase chain reaction, that is PCR. But it is cell-based cloning which has been a real favorite of biotechnologists, in which in a cell that is quick to multiply and a cell that can harbor a foreign piece of DNA is first transferred with a foreign piece of desired segment of DNA. Let's say that we have transferred a gene into a host cell, and after that, once we have established that the host cell has taken up the gene of interest, we culture the host cell for several generations, and then we get a really large number of host cells, and each one of these host cell harbors the gene of interest. That is cell-based cloning. Cell-based cloning is one of the big methods how gene cloning is done, and this is where I am dropping another clue, that cell-based cloning gives rise to the gene library and genome library. So cell-based cloning you have to study in detail, and after that you will be going with the basic method of cell-based cloning and the role of vectors in the entire process. Once you have developed this entire understanding on DNA cloning, you are actually good to go to the next topic, that is recombinant DNA technology. Recombinant DNA technology, in my opinion, is a slightly larger topic where there are a number of small topics, plus there are a number of hidden topics also. First and foremost, like any other topic, you will start with a good definition of what is a recombinant DNA, what kind of DNA we call recombinant DNA. Again, unfortunately, most of the common resources easily available on internet, they will be unable to give you a concrete and irrefutable understanding of what is a recombinant DNA. And I'm repeating myself that there is never a substitute to read a good book or be taught by a good teacher or be reliant on a good material. Anyways, so find for yourself what kind of DNA will be called correctly recombinant DNA, and then what is that umbrella technology in which there are so many techniques which come together to give rise to this technology called recombinant DNA technology. This technology is about identifying the DNA of interest, isolating the segment of interest, combining it with the vector, transferring that vector in the right host cell, screening the right host cell to actually establish the acceptance of the DNA, and finally culturing the host cell to get the desired result, which may be cloning the inserted copy of gene into many copies or getting the protein product of the inserted gene. Whatever it is, these are the steps of recombinant DNA technology. So define both of these things, recombinant DNA and recombinant DNA technology, before moving further. So once you have done that, get into the history of that. The recombinant DNA technology was established as a technology by Stanley Cohen and his co-workers. They all are Nobel laureates, so it has been a significant achievement in the field of biological science. You need to know who were the people behind the earliest experiments of recombinant DNA technology, and one name I have already given you, that is Stanley Cohen. You have to find his co-workers' name, that is Herbert Boyer and Paul Chang. And then there were other scientists also. Mention the year and also mention what did they create out of their first experiment. It was actually a recombinant plasmid that we call the PSC family plasmid. So what was the earliest PSC family? P stands for plasmid, SC stands for Stanley Cohen, and then there is a numerical designation given to this earliest developed plasmid. So this much of history you certainly need to know, and also you need to mention certain enabling technologies. For example, recombinant DNA technology would not have been a reality had restriction endonucleases not been discovered as a class of enzyme. Because without restriction endonucleases, you cannot cut the DNA at a precise location, and if you cannot cut it, how do you create two different combinations of or multiple different combinations of DNA? So here you are going to discuss the contribution of Nathan Sarber and Hamilton Smith, who were essentially the discoverer of recombinant DNA technologies, molecular seizure, that is restriction endonucleases. Know all about the history. This is going to enrich the quality of your answer, especially when you are writing a general answer on this. After that, get hold of the tools. First, for cutting DNA, it is restriction endonucleases, but again, restriction endonucleases fall in three classes. Class 1 and class 3 do not have much use, but it is class 2 which cuts very precisely, plus there are other properties also of class 2 which makes it a little more useful to recombinant DNA experiments. You need to know all about restriction endonucleases, and here I must say that on restriction endonucleases you can expect a standalone short note, so you need to cover this topic a little well so that you can write a short note on restriction endonucleases. Coming to the main list of topics, then how do we separate the DNA? We separate the DNA by subjecting them to electrophoresis. Gel electrophoresis is anyway a topic to be studied in paper 1 bioinstrumentation. Why not study gel electrophoresis here only and make use of this understanding in bioinstrumentation later? So gel electrophoresis, how do we calibrate the gel? How do we run the gel? So on, you need to know all of this. After that, identifying the DNA. In a mixture of DNA of different lengths and all that, how do we finally isolate the DNA that carries the sequence of our interest? There are probes for that. Probes are single-stranded nucleic acids with complementary sequence to the DNA segment which we are actually trying to search. So know about the probes, and that will give you an understanding how do we identify the DNA. Ligating the DNA is carried out by certain types of ligases. These ligases can be the standard ligase, or they can be ligase obtained from T4 bacteriophage. Under what circumstances we use the T4 bacteriophage is something that you should be exploring. Lastly, what are the modes of a screening once we have transferred the DNA of interest? Mode of a screening means that once we have conducted our DNA transfer experiment, we would like to make sure that the host cell where we were trying to transfer it has actually taken it or not. There are certain steps. How do we carry out the screening? You need to know, and this is where you are going to familiarize yourself with the concept of selectable marker and scorable marker. In this, also try to know what is a reporter gene. After you have done all of these, you will be coming to the method. There are two basic approaches to carry out recombinant DNA experiment. First, you work right from the beginning with a known piece of DNA segment. Let's say that you want to transfer human insulin gene into Escherichia coli. So you can start with the RNA of human insulin, and you will be reverse transcribing this RNA, and you will be creating complementary DNA or cDNA. After that, this cDNA will be made double-stranded, and right from the beginning you are knowing that you are working with the desired segment. So this approach is known as a screen and clone. In the beginning only you carry out your screening and then you begin the cloning. But the second and more commonly used approach of 1990s was a clone now, a screen later. It is also known as the shotgun approach of gene cloning. Know about both the methods and how do we go about it, and then individual steps need to be done in detail. How do we carry it out? How do we get the gene of interest? How do we choose the vector? What are the considerations when we are choosing the vector? It is the host cell, it is the side of the genetic material that we want to transfer, it is the presence of markers, it is the nature of the multiple cloning site or polylinker site. All of these play a big role in selecting the vector. In this step, know about vectors in detail, because the very next topic is all about vectors, especially of the three categories: plasmids, cosmids, and artificial chromosome. So why not educate yourself on gene vectors? What are gene vectors? What are their desirable properties which makes them a good gene vector? And then how do we choose the gene vector? So on. After that, you will create a recombinant vector or R vector. A recombinant vector is the vector which has gotten the gene of interest which you wanted to ligate into the vector, and then using this recombinant vector you will be trying to send the gene of your interest into the host cell. All of this is creating the recombinant vector. Here you cut the vector, with a specific restriction enzyme, you try to cut the donor DNA segment with the same enzyme, and using the complementary sticky ends, you try to ligate them. But if that is not a possibility, you will be going for blunt cut, and even if that is not a possibility, you can overcome the mismatch using the adapters, so on. You need to know all about this. After that, you will be carrying out gene transfer. The method of gene transfer varies what kind of cell you are trying to transfer your gene into. Bacterial cells will be responding to different modes of gene transfer, animal cells will be responding to different modes of gene transfer, and plant cells will have their own unique set of tools to carry out gene transfer. At least you need to know very well about the bacterial cell and animal cells. That will give you a good coverage of how do we transfer the genes. Lastly, you will be coming to the screening. How do we screen the cells whether they have got the gene of our interest or not? Once you are done with this, so you have actually covered the entire method very, very well. And then it is the application part. What are the applications of recombinant DNA technology? Because there are so many applications of recombinant DNA technology, it will be a very good idea to categorize the application areas. For example, how do we use them in the field of medicine? How do we use them in the field of environmental cleanup? How do we use them to improve the quality of farm products? How do we use them to improve the quality of farm animals? So on. And then under each category, you will be discussing individual cases. In just one sentence, you will be mentioning one example, and that will give you a real wholesome list of application areas of recombinant DNA technology. Once you are done with all of these, you are really safe with recombinant DNA technology, and then you can take a deeper dive into the field of vectors. So moving to the next topic, that is of vectors. So this is what we have to study. But I'm presuming that you have already done the basics of vectors, the properties of vectors, what makes a good vector, how do we create vector and all that. So you can follow one common format for these three categories of vector. But please understand that for each vector, a separate question can be asked to you. Besides, you can be asked to compare it. So be ready for two types of question. First is standalone question. A standalone question will be a question directly on a vector or a question based on comparison. So these are the two types of question that can be asked. But if you study each vector under the same set of subtopics, it will be a good coverage and it will allow you to answer either a standalone or a comparison based question. So first and foremost, what is this vector? How do you define a cosmid? So cosmid is the cos sequences combined with a plasmid. What is a cos sequence? It is a cohesive sequence. Where do we find it? We find it in lambda bacteriophage. So that way you go about building a cosmid. Similarly, what is an artificial chromosome? What are the elements of bacterial artificial chromosomes? What are the elements of human artificial chromosomes? And what are the elements of yeast artificial chromosomes? After that, how is this vector constructed? What are the elements in this vector? And how do we go about combining these elements? Then what is the final structure of the vector? What are the parts of this vector? You need to draw a neat line diagram and show the part of the vector and at the same time describe the parts of the vector. Then you come to the diversity. For example, there are so many types of plasmid vectors, there are so many types of artificial chromosomes, and there are many types of cosmid vector. Essentially, plasmids, cosmids, or artificial chromosomes are classes of vectors, but within these classes there are many individual types of vectors. You need to know the important examples here. After that, in what kind of rDNA experiment, recombinant DNA experiment, are we going to use these vectors? That will be an important point to study. After that, what are the steps of using it? How do you isolate and ligate the gene? How do you try to transfer this vector, etc. For example, cosmids can be transferred using a virus, whereas plasmids can be transferred generally using co-cultivation or membrane permeabilization, etc. Then what are the benefits and successful examples of using this vector? And lastly, what are the limitations of an individual vector? Each vector has its own limitation. Had there been no limitation, one vector would have been enough for the entire range of recombinant DNA experiment. Since that is not the case, it is obvious that there are going to be limitations of individual vector. You will be knowing that. Once you are done that, you are completely done with the recombinant DNA, cloning, and all about vector. Now you can come to transgenics. First, let's get very clear about what is a transgene, what is a transgenic, and what do we mean by transgenics. So transgenics is that branch of biological science which deals with transgenic organisms. So it is concerned with production of transgenic organisms, studying their properties and further manipulating them. Transgenic animal is an animal that carries a transgene stably. That means it can pass the transgene to its progeny, and at the same time, if it is meant to be expressed in the host animal, it expresses the transgene. So what is a transgene? Transgene is a gene that is obtained from member of another species. So that way, sequentially, first what is a transgene, what is a transgenic animal, and then finally what is the discipline of transgenics. Get hold of these definitions. As mentioned earlier, a good definition is an asset that you can use at so many other places within your zoology paper. After that, get to know the basic history of animal transgenics. Who were the pioneer, what were the experiment they conducted. These are important things which you will be needing while writing a general answer. Then you will be coming to the scientific core of the topic. What is the rationale of animal transgenic? What are the reasons why the scientists have been interested? And understand that commercially important product is just one of the rationale. Initial development of transgenic animal was primarily driven for research purposes. What were those research purposes? You will need to know. Then, what are the methods? How do we transfer a foreign gene of interest into an animal? Do we manipulate the gametes? Answer is yes. Do we manipulate the early pro-embryo? Answer is yes. Do we manipulate a little late embryo, say the embryo that is in pre-blastula stage? So the answer is yes. Even pre-blastula embryos are manipulated. All of these are different methods of producing transgenic animals. So in transgenics, you will be dealing with how do we transfer the gene of interest, and after transferring the gene of interest, how do we raise a transgenic embryo, and then how do we check the stable integration of the foreign gene, and how do we check the stable expression of the foreign gene. All of these will constitute the steps of a transgenic experiment. Once you are done with that, you have covered the scientific core. After that, what are the beneficial examples? Do we have lambs which give us better quality wool? Do we have fishes which give us greater yield? Do we have farm cattle which are resistant to disease? Do we have pigs which actually carry a foreign gene and they contribute very less to the environment pollution? Do we have cows which give milk less in lactose content so that people who are lactose intolerant can still safely consume this milk? Do we have cows which give milk with certain vaccine properties? Answer to all these questions is yes, we do have these animals, and these will be your examples of beneficial applications of transgenic animals, and then you will be talking about its problem areas. EELSI. First E is for environment, second is for ethics, L is for legal, S is for social, I is for issues. So when we go with transgenic animal formation, there are going to be environmental issues, there are going to be ethical issues, legal issues, and social issues around this, and you have to deal with each one of these issues before you can claim success with transgenic animals. You need to do all of these, and the application of beneficial in the beneficial direction is going to form the core of your answer on transgenic animal when it comes under economic zoology in paper 1. Now we can move to the next topic. So what is the next topic? The next topic is whole animal cloning. This is again a little big topic, but what are the sub areas that you have to do in whole animal cloning? First and foremost, understand through a good definition what is a clone and what is cloning. It is not so easy to define a clone. So try to define what is cloning and what is a clone, and try to find the different levels of clone. So this is again a small challenge that I am giving to you. You don't need to tell me, but you certainly need to explore the real meaning of clone. Do not look for these answers on the internet. Go for a good textbook and you will get a good answer. So after that, whole animal clones, you need to know a brief history, how the first clone was produced, and then what were the major landmarks, what were the works of Ian Wilmot, which got worldwide attention, and which actually made cloning a hot topic. And it was actually after Ian Wilmot's experiment and development of the first somatic cell nuclear transfer based clone that made cloning come in the biological and general discourse both once again, and after that only it was added to your syllabus. So a quick history will be very beneficial. Then what are the types of whole animal clones? So first types is twin, essentially created by a splitting of the zygote, and after that the second type is nuclear transplant based clone. There are two subtypes. One is embryonic nucleus transplant, which is something that we have been doing since early 1980s only, and then we have got somatic cell nuclear transfer method. Somatic cell nuclear transfer method is actually a breakthrough which was never attempted before Ian Wilmot. When Ian Wilmot approached this, he had a very high failure rate, but eventually he was able to produce a female sheep called Dolly. So after its birth, it was called Dolly, and that made this topic explode in the popular discourse, and that raised the debate whether cloning is ethical or not, all of this. But in this topic, you do not have to go into all those debates. You have to focus on the scientific core. After that, you will be dealing with how we produce twins if we are going to produce it artificially for an animal, and then how do we go about nuclear transplant based method. First, embryonic cell nuclear transplant, and second, somatic cell nuclear transplant. And then get into the details of somatic cell nuclear transplant, because this was the method which was revolutionary when it was first attempted. Now it has become a little common technology, and a lot of labs, including labs in India, for example NDRI at Karnal, Kashmir University of Science and Technology, all of these have carried out different types of variants of somatic cell nuclear transfer, and successfully so. So you will be actually going a little deeper into this. You will be talking about its steps, why it is such a revolutionary method, and also what are the limitations. Then how are we conducting cloning in India? I mentioned Kashmir University of Science and Technology, Agriculture Science and Technology, it is SKUAST, and then NDRI Karnal, and there are a lot of other labs which are into whole animal cloning. What is therapeutic cloning? To give you an idea, therapeutic cloning is where somatic cell nuclear transfer cloning meets stem cells. Prepare a short note on this, prepare a short note on cloning in India, prepare a short note on this, and this overall itself is a question. So these are the potential questions here in cloning, and then talk about the benefits and limitations of cloning. When you are talking about the benefits, do not write imaginary benefit, write the real benefits which have already been established by the scientists. The next topic is RFLP, RAPD, and AFLP. These are again the topics which are related to different molecular marker tools, and same format can be applied to each one of them. First and foremost, what does the term mean? For example, RFLP stands for restriction fragment length polymorphism. So you need to write it in full, and then you need to explain in simple English what essentially it means. Then what is the brief history? Who developed this method? It might be a lab, it might be a scientist. So when was it developed, and what was done with this? So that is a brief history. What is the underlying principle? Why restriction fragment length polymorphs arise in the first place? So that underlying principle needs to be mentioned, and then come to methods and steps. If we are trying to have an idea of restriction fragment length polymorphism, how are we going to go about it? How do we process the DNA? How do we run the DNA on the gel? How do we identify the DNA? How do we create the blots and establish the polymorphism in the length of restriction fragment? So that is the methods. After that, what are the application areas of this technology? Finally, benefits and limitations. What are the benefits and limitations of using RFLP? What are the benefits and limitations of using AFLP? How rapid RAPD is applied and what are its limitations? And also be ready. So each one is a question. RFLP is a question in itself. RAPD is a question in itself. AFLP is a question in itself. Apart from that, you can be asked comparison questions also. So be ready for the comparison question as well. Then RFLP and its application in DNA fingerprinting. The syllabus writes DNA fingerprinting. Modern day scientists they prefer to use DNA profiling. So because you have already studied RFLP earlier, so your job is going to be simpler. So here, what is RFLP? How RFLP, that polymorphism arises, what is the underlying principle? Presumably you have already studied that before coming to this topic. And what is the principle for RFLP being applied for DNA profiling? So how can we harness RFLP to profile DNA samples? Then a quick history of DNA profiling, that who was the scientist who developed the DNA profiling method, so on. And after that, learn in detail the Southern blotting based method of DNA profiling. Once you are done with that, you should know a basic idea. So in brief, you need to do other methods of DNA profiling, because RFLP characterization is not the only mode of DNA profiling. There are other modes also. What are the application areas of DNA profiling? What are the benefits? What are the limitations? And then from your GS preparation, you have to import India's DNA profiling law. So India is having a law on DNA profiling. What are the main provisions and what are the main objectives of that law? You need to know. So that will be all about this, and then you can study genomics. So genomics, what's a genome? Again, a clear definition is mostly missing. When I speak with my students before teaching this topic, I see that most of us have a little confused understanding of what is a genome. You do not need to be one of them. You need to know very clearly what is genome, and then what has been the history of genomic studies. And in this, you are going to pay attention to the method of Fred Sanger, who for the first time sequenced the genome of this virus 5X174. So this was the first ever genome to be sequenced, and that was done by Fred Sanger. Fred Sanger is a two-time Nobel Prize winner, first for establishing how do we sequence protein and later for establishing how do we sequence DNA. So Fred Sanger's method is known as dideoxy chain termination method. You need to know the details of this method. After that, what are the steps of genomic studies? How do we isolate the genome? How do we cut the genome into smaller fragments? Why at all do we have to cut the genome into smaller fragments? How do we go about sequencing of the individual fragment? And then how do we ligate the data of individual fragment sequencing? So there is a method that is called contig by contig, and then there is a method that is called shotgun approach. So what is the shotgun approach and what is the contig by contig method? You need to know both of these. And once you are done with this, then what are the application areas for genomic studies? That will be quite a good and sufficient coverage of genomics. And also be ready for a comparison on genomics and proteomics. So it's been asked in the syllabus also, they have been mentioned together. So a short note, comparison on genomics and proteomics, and then genomics is a standalone, is a question for you. After that, the Human Genome Project. This is going to be easy because you have already studied what is genomics. So here, overview, what was Human Genome Project, etc. then what were its objectives? So Human Genome Project did not have objective of simply cloning the human genome, simply sequencing the human genome. It had a number of other objectives. When you read it, you will find that. So it worked with 6 different objectives. Then what was the history and what were the landmark development? Right from Human Genome Organization formation under the leadership of Dr. James D. Watson until the HapMap project, you have to know all the landmark developments. And after that, what was the approach to Human Genome Project? So Human Genome Project was one of the biggest collaborative biological science research project which was launched at its time, and so many labs from different parts of the world were collaborating, but collaborating in a very systematic manner. So what was the approach? Later on, how did the private sector player come to play a role, and especially the role of cellular genomics? After that, what were the key findings of the Human Genome Project? You need to have a list of 10-15 points. What are the application areas of Human Genome Project finding? How does it promise to revolutionize the field of medicine, the field of disease diagnosis, disease prediction? How does it pave the way for molecular medicine, so on? What are the ethical, legal, and social issues arising from Human Genome Project? And finally, what are the further projects which are being planned or which are already being carried out after the Human Genome Project? So on Human Genome Project, you have generally a direct question, plus there has been a short note on findings of the Human Genome Project and its applications. So these two have also been asked as a short note. Next one is the Ribozyme Technology. So ribozymes are RNA enzymes. They were discovered independently by Sidney Altman and Thomas Cech, and they for the first time realized that even certain RNA can be catalytic in the biological context. Today we know that the ribosome enzyme peptidyl transferase is an abundant ribozyme. So what are ribozymes? Who discovered, and what was the example that they discovered, and what are the characteristics of ribozyme? Here you are going to discuss the physical as well as the structural characteristics of ribozyme. Then what are the features because of which we can use them for human welfare? So the essential thing is that most of the ribozymes are good at targeting other RNA, cutting them, destroying them, modifying them. So this is what most of the ribozymes tend to do. And if this is the case, why not we use ribozymes to destroy the RNA for such genes, such mutant genes whose expression causes different types of human diseases, including cancers, including autoimmune disorders, so on. So these are the certain underlying principles, but of course you need to study more to get more points. And then what are the approaches and methods to use ribozyme? Give example. Finally, application areas and success so far, and what are the limitations of ribozyme? Generally, a single question: what is ribozyme technology? Discuss its methods and application. This kind of question has been asked in the examination, and I personally feel that it is this kind of general question which will be asked on this topic in future also, because going deep into the topic will actually getting too deep from the student's perspective. So on this, just prepare a good short note in 2 or 3 sides of an A4 sheet, and that will suffice. Last topic is proteomics. Proteomics is protein equivalent of genomics. So when we talk about a cell, so all the proteins expressed in a particular cell is called the cellular proteome. So when we study proteins of a given cell, given tissue, or a given organism, this branch of science is known as proteomics. Proteomics is basically the study of genes product. Majority of the genes are protein encoding genes. So merely having a gene does not make you predisposed to certain diseases or certain condition. The question is whether that gene is expressing or not. How do you establish whether the gene is expressing or not? It is by proteomics. So proteomics is an important discipline of applied biology, which helps us in molecular medicine, which helps us in establishing the predisposition to diseases, so on. So what is a proteome and what is proteomics and why proteomic study is important? Once you are done with that, you will come to the method, the 5 steps to analyze protein sequence. First, you go for sample preparation, you separate the protein, you ionize, and then you go for mass spectrometry, and then establish the sequence information and organize it. So these are the 5 steps to establish the sequence of protein. After that, you go about protein comparison. How do two proteins from two different organisms or within the same organism compare to each other? Protein-protein interaction, because a large number of proteins do not act in isolation, they act as a part of a complex. So protein-protein interaction is again a very important part of proteomic studies. And lastly, you will be studying protein families and their evolutionary relatedness. All of this in proteomics. After that, what are the application areas and what are the limitations of proteomic studies? These are the things that you have to study in proteomics, and this was the last topic of our applied genetics part. Now, in the end, as I was telling you, that standard books of genetics will not be very good so far as applied genetics is concerned. So my favorite book here is on the left. It is by Old, Primrose and Twyman. So this is an excellent book. Principles of Genetic Manipulation and Genomics. If you have time, there is no book which comes parallel to this. So this is my personal favorite. But if you are more used to of reading an Indian book, then Dr. P.K. Gupta's book on biotechnology is a very readable book. I would rate it among the best written by an Indian author. And if you want to kind of save time and have a concise exam-oriented coverage, then there is nothing to beat the evolution study material, which I have compiled, edited, and made it the UPSC compatible notes. So whatever is your choice, you can go ahead with this, stick to your choice, make good self notes, and you will be sailing through in the examination. So on that note, I will take your leave. Stay consistent with your studies, and it will be giving you excellent results in not so far future. All the best and good luck with your studies. Stay safe.